The overall goal of the following experiment is to detect changes in alternative splicing during EMT. This is achieved by utilizing an inducible EMT model, in which expression of the fusion protein Twist-ER in the human mammary epithelial cells triggers cells to undergo EMT upon tamoxifen treatment. As a second step, the expression of splice isoforms is analyzed by quantitative RT-PCR using isoform-specific primer sets, which reveals alternative splicing changes at the RNA level. Next, the abundance of protein corresponding to each isoform is determined by immunoblotting in order to confirm the expression of splice isoforms at the protein level. Results are obtained that show changes in alternative splicing in genes of interest during EMT based on quantitative RT-PCR and immunoblotting analyses.
This method can help answer key questions in the field of RNA alternative splicing and EMT, such as how alternative splicing is regulated during EMT, and how this regulation affects EMT and EMT-related pathological processes.
The implications of this technique extend towards cancer therapy because EMT plays an important role in promoting tumor invasion and metastasis.
Though this method can provide insight into alternative splicing during EMT, the method for isoform-specific mRNA detection can also be applied in other systems, such as studying alternative splicing in